Welcome back to the Deep Dive, where we take complex, cutting-edge research, synthesize a stack of your sources, and really try to extract the most important nuggets of insight. Now let's talk about the most fundamental biological process of all, breathing. We inhale oxygen, and it fuels our every move, our every thought. It makes high-energy, complex existence possible. But what if the source material we've been given today, a deep dive into the evolutionary history of aging, titled The Oxygen Tax, Evolutionary Origins and Reversing Biological Decay, suggests a radical, completely counterintuitive truth?
It really does.
Our sources argue that death, specifically biological decay and senescence, it's not an unavoidable law of physics. It isn't a design feature of life. It is quite simply a catastrophic, multi-billion-year-old evolutionary compromise.
A compromise, that's exactly the right word.
We're diving into the surprising reality that oxygen is simultaneously the highest grade fuel that granted us consciousness and the relentless molecular poison that guarantees our decay. Our mission today is to thoroughly unpack this foundational bargain and look at the molecular tools being developed to maybe, just maybe, undo it.
And that mission requires a lot of intellectual precision. We aren't just summarizing biogerontology findings. We are tracing mortality all the way back to its molecular origin story.
Right.
We need to understand the path from simple life that exhibited what you could call functional immortality to, well, to us, to the current high-energy, complex human existence. We will trace this path from the most dramatic climate event in Earth's history, the Great Oxygenation Event, which really initiated this whole toxic endeavor right through to the modern, aggressive promise of overcoming what evolution left behind. Specifically, this concept known as the selection shadow.
Okay, so we're connecting ancient history to modern cell failure.
Exactly. We'll be connecting the dots between ancient history and the highly specific forms of modern cellular failure, examining the exact waste products that are the molecular currency of this oxygen tax. Things like reactive oxygen species, the accumulating lipofuscin, and the stiffening advanced glycation end products. And critically, we have to spend serious time analyzing the why, the strategic dilemma that allowed this decay to persist, which is all codified in the disposable soma theory.
Okay, let's unpack this. Before we get to the high-tech fixes, we have to start with a journey way back in time to a period when death, wasn't guaranteed. We are talking about rewinding the clock 2.4 billion years before the air itself became toxic. Imagine the Earth in this deep past. The atmosphere was radically different, rich in methane, largely devoid of oxygen. Life was simplistic, primarily single-celled prokaryotes, and overwhelmingly anaerobic.
Right, no oxygen.
And this life existed in a state that researchers call functional immortality. This concept is challenging to grasp because decay is so central to our current biological reality. But the source material explains the core mechanism brilliantly. Simple binary fission.
It's a very elegant system.
In binary fission, the process is one of simple duplication. The cell copies its contents and divides exactly in half, yielding two seemingly identical daughter cells. The critical, defining detail here is that the lineage is a continuous, self-renewing loop.
No parent left behind.
Exactly. When we, as complex organisms, reproduce, we leave behind a parent body that continues to decay. These ancient cells didn't. The body of the parent literally becomes the bodies of the children, and in doing so, they avoid the accumulation of time-related systemic defects. There is no biological parent left behind to get old.
That continuous lineage is, for all intents and purposes, the biological default. It establishes the baseline that aging is not mandatory. If we define aging as the internal systemic accumulation of irreparable damage over time, the mechanism of binary fission, by definition, prevents it.
Okay, but it can't have been perfect, right, even back then.
That's a great point. And to maintain intellectual rigor, we have to introduce a nuance that the sources meticulously lay out. Even this primitive life wasn't perfectly immortal, and it developed the first simple evolutionary strategy against accumulating tribe asymmetric division.
Asymmetric division, so not a perfect split?
Not always, no. This is often dubbed the old pole problem. When the cell prepared to divide, the splitting wasn't always perfectly symmetrical in the distribution of cellular materials. Research shows that one daughter cell would frequently inherit the older portion of the cell wall structure, the old pole, and a disproportionate amount of accumulated, misfolded, or damaged protein aggregates.
So it was deliberate, not a mistake.
It seems so. It wasn't a flaw, it was a strategic choice by the lineage.
So effectively, the cell designated one half of the division as the sacrificial trash collector.
Precisely. It functioned as a primitive waste management system for the overall genetic line. By concentrating the internal waste, the minimal damage accumulated even under anaerobic conditions, into one recipient cell, the other daughter cell could start its life effectively pristine.
which would let it grow faster.
Exactly. It could grow faster and reproduce more efficiently. The burdened old pole cell would typically exhibit a slower growth rate, face reduced function, and might eventually perish. But the lineage as a whole, the clean, fast-growing line, maintained its youthful vigor.
That's fascinating. It's like planned obsolescence at the cellular level, but for the benefit of the line, not the individual.
That's a really good way to put it. And this point is crucial because it shows that even before the introduction of oxygen, life had to contend with entropy and waste, but its answer was lineage-based. Death in this era, as the source notes, was overwhelmingly accidental.
Right.
It wasn't the result of an intrinsic programmed clock running down. It was based entirely on external environmental factors.
So starvation, chemical poisoning, or just being physically destroyed. There was no genetic countdown clock tied to passing time.
None at all.
This functional immortality, coupled with that primitive waste dilution strategy, proves that decay is not an inevitable prerequisite for life itself. It is a biological calculation made later in history, a calculation driven by the need for massive amounts of energy to support greater complexity. So now we make the jump forward to the massive inflection point, the Great Oxygenation Event. Suddenly, the metabolic waste product of simple cyanobacteria oxygen flooded the atmosphere.
A global pollution event, really.
Absolutely.
Yeah.
And to the anaerobic life that had thrived for billions of years, this gas was, quite literally, a potent poison. It was highly reactive, cytotoxic, and forced an extinction-level environmental crisis.
It was a crisis that demanded a radical solution.
And the survival strategy that emerged roughly 2 billion years ago was one of the most radical and defining events in all of evolution. This was endosymbiosis, where an anaerobic host cell essentially engulfed an aerobic bacterium. Instead of digesting this bacteria, the host cell integrated it, transforming it into the modern mitochondrion. This merger established the eukaryotic cell, the basis for all complex life, including us.
And that, despite the initial toxicity, was the ultimate energy bargain. The payoff for hosting this high-performance aerobic engine was astronomical. The sources highlight a critical comparative advantage: a 15-fold increase in adenosine triphosphate ATP production compared to the older fermentation-based anaerobic methods.
15-fold, that's a staggering leap.
It's a game changer. This exponential surplus of energy was the evolutionary currency that paid for everything: complex mobility, large-scale multicellularity, specialized organs, large brain size, and ultimately, sentience.
But there's always a cost.
Always. The evolutionary price tag for this energy was the unavoidable toxic legacy, what the sources call the oxygen tax. If you are constantly processing oxygen for energy, you are effectively running an extremely hot chemical furnace inside the cell, and that furnace inevitably leaks. This is where the technical detail is essential for an informed listener to really get it.
We talk about reactive oxygen species, or ROS, all the time. But how exactly does the leak happen at the molecular level within the mitochondria?
Okay, so the ROS leak is a fundamental flaw in the electron transport chain, or the ETC. The ETC is designed to pass electrons down a gradient to eventually meet oxygen, generating ATP. It's like a tiny biological production line.
But it's not perfect?
Not even close. It's not 100% efficient. At complex I and complex 3 in the inner mitochondrial membrane, electrons sometimes escape and react directly with oxygen, prematurely creating the highly unstable superoxide radical.
And that's the first domino to fall.
That's the first one. This superoxide is then rapidly converted into other, even more destructive radicals, notably the hydroxyl radical, which is just notoriously reactive. These ROS species desperately seek stability by stealing electrons from essential cellular components.
And that's the corrosion.
That is the mechanism of the corrosion. They cause single-strand breaks in DNA, they induce lipid peroxidation, which is like turning healthy cell membranes rancid, and they indiscriminately misfold and damage proteins. They are truly biological rust that attacks every major class of molecule in the cell.
So the very engine that powers our higher functions is simultaneously degrading the hardware upon which those functions rely.
Yeah.
This is the definition of a stark existential trade-off.
A stark trade-off is right. And the problem doesn't stop at simple corrosion. The sources detail a critical systemic consequence of the mitochondrial legacy.
Inflammaging.
Inflammaging. So inflammation and aging combined. Exactly. It's this chronic, sterile, low-grade systemic inflammation that acts as a powerful driver of accelerated aging across the entire body, from vessel stiffness to neurodegeneration. The mechanism behind inflammaging is fascinatingly evolutionary. Remember, the mitochondrion is, genetically and structurally, an ancient bacterium that was engulfed.
Right, the endosymbiont.
So when these tiny power plants get damaged or stressed by ROS accumulation, their contents and molecular signatures leak out into the cytoplasm. Our immune system, specifically the innate system, recognizes these leaked components not as waste, but as damage-associated molecular patterns or DAMPs.
It sees them as a threat.
It sees them as an invader. A prime example is cardiolipin, a phospholipid usually sequestered to the inner mitochondrial membrane. When cardiolipin leaks, the cell's internal security system, the inflammasome, interprets this specific signature as an ancient bacterial invasion.
It's an immune ghost from 2 billion years ago.
Exactly. It's a perfect description. This interpretation triggers a defensive, inflammatory response that, while brilliant for fighting an acute infection, becomes chronic and disastrous when applied to the slow, relentless accumulation of internal mitochondrial exhaust over decades. We are perpetually running a low-level, self-destructive inflammatory state because our own power plants still look like foreign invaders to our security systems. And this chronic inflammation, this inflammaging, it just accelerates systemic aging across all tissue types.
This molecular history leads us to the central, necessary question raised by the sources. If the energy advantage is so vast, why hasn't natural selection engineered a maintenance system robust enough to fix the inevitable rust? Why is mortality the default for complex multicellular life?
The answer is pure evolutionary pragmatism, and it's completely devoid of sentimentality. It is entirely dictated by energy and resource efficiency. It is encapsulated in the disposable soma theory, which is a foundational concept in biogerontology.
The disposable soma.
Yes. This theory posits that the body, the soma, is treated by evolution as nothing more than a temporary expendable vehicle.
A vehicle for what?
A vehicle for the genes. Its only purpose is to successfully house, protect, and propagate the germline, the genetic material. Once an organism has successfully transferred its genes to the next generation, the biological pressure to maintain that specific vehicle drops immediately and dramatically. Evolution is concerned with genetic success, not individual permanence. Any resources spent on maintaining a flawlessly repaired body after successful reproduction are, in evolutionary terms, wasted energy that could have been better spent on ensuring the survival and fitness of the offspring.
So we're not built to last because maintaining perfection is just too expensive once the primary evolutionary mission reproduction is complete. It's a calculation of maximizing energy return on investment.
Precisely. And the key mechanism driving this acceptance of decay is the selection shadow. This is a powerful, almost philosophical concept. Natural selection acts as an intense filter, but its power is heavily focused on the early stages of life.
So it's strongest when you're young.
At its absolute strongest. Selection acts with maximum force on traits that appear before or during the reproductive window, ensuring survival to reproductive age. But once genes are successfully passed on, the evolutionary pressure simply vanishes. Any genetic mutations or accumulation of metabolic waste products that cause damage in late life, say, after the ability to reproduce effectively ceases fall outside the shadow.
They're invisible.
They are no longer selected against because they do not impede the successful transmission of genes to the next generation. For evolution, late-life decay is genetically invisible. The body essentially gets a biological yellow light once fertility is achieved.
So if a molecular failure causes neurodegeneration at age 85, evolution simply shrugs because the line has already propagated. But this seems to imply a conscious choice to redirect resources. How does evolution actively enforce this decay rather than just ignoring it?
Because resources are always limited and repair is metabolically expensive. It's all about budgeting. Evolution strategically diverts energy away from the high-cost metabolic activities required for flawless cellular repair, the systems that would fix mitochondrial damage, and prioritizes the immediate critical needs of fertility, survival in youth, and reproduction.
It's a trade-off.
It is a fundamental energy budgeting decision where maintenance budgets are systematically slashed post-reproduction, and this point is compounded by another genetic phenomenon cited in the sources. antagonistic pleiotropy.
Okay, that sounds complicated.
It's a bit of a mouthful, but the concept is straightforward. This is where a single gene offers a genetic double-edged sword. It provides a massive, overwhelming survival and reproductive advantage in youth, but has destructive side effects that accumulate unnoticed in later life.
Can you give us a concrete example of a gene that demonstrates this kind of trade-off?
Absolutely. Think about genes that regulate inflammation or cellular proliferation, like the p53 tumor suppressor pathway. P53 is crucial for youth survival. It rapidly triggers cell death or apoptosis in damaged cells, which suppresses early cancers and maintains tissue integrity.
So it's a huge early life advantage.
A huge one. However, sources suggest that high activity of P53 across a lifetime contributes to the depletion of stem cell pools and chronic low-level inflammation, accelerating aging phenotypes later on.
So it's good early, bad late.
Exactly. Evolution tolerates this long-term cost because the immediate reproductive benefit cancer suppression in youth far outweighs the distant post-reproductive cost of accelerated senescence. It is a cold, hard trade of longevity for immediate vigor.
Let's focus this strategic dilemma now onto the highest cost, most vulnerable parts of the human organism, the cells that cannot divide. These specialized cells represent the ultimate biological expression of the oxygen tax, and they are the bottleneck to extreme human longevity.
They really are the Achilles heel.
We are primarily talking about two critical cell types: the neurons in the brain, which govern our consciousness and memory, and the cardiomyocytes, the muscle cells that drive our heart. They are our most advanced components, but also our weakest link, because they are fundamentally post-mitotic.
And their post-mitotic nature is the crux of the identity trade-off. This lack of division isn't a flaw, it's an essential requirement for stability. For you to maintain a consistent self, your neural wiring, the intricate network that stores long-term memory, must be fixed and stable.
Right, you can't just be rebooting your brain all the time.
You'd lose your memories, your personality. If neurons were constantly dividing and reshaping themselves, like skin cells or blood cells, that stable neural configuration necessary for personal continuity, your identity would be erased constantly. And the heart requires the same stable connectivity for reliable, rhythmic contraction.
Okay, so stability is essential, but what's the cost?
What's fascinating here is the unavoidable consequence of that stability, the profound inability to dilute trash. In every other cell that undergoes mitosis, the accumulated mitochondrial waste is split and shared between the two daughter cells during division, effectively slowing the rate of damage accumulation for the genetic line.
But neurons and heart cells have to hold on to literally everything they produce or fail to recycle from the day they are fully differentiated until the day they die.
For their entire lifespan. Decades. They are forced to retain the complete lifetime of mitochondrial exhaust, the ROS damage, and all the ensuing metabolic byproducts. They are the ultimate victims of the oxygen tax, and this retention leads to the decades-long buildup of specific, problematic waste products.
And the sources get specific about what that waste is.
They do. Let's start with lipofuscin. This is often described as the wear and tear sludge or age pigment. It's a pigmented, insoluble aggregate composed of oxidized lipids and proteins, particularly those damaged by ROS.
So it's just junk.
It's junk that accumulates inside the cell, specifically within the lysosomes.
The lysosomes are the cell's recycling plants, right?
Exactly. They are supposed to break down and recycle cellular junk. But lipofuscin is indigestible. The lysosomal enzymes cannot break down the complex cross-linked aggregate.
So the recycling plant gets clogged.
It gets completely overwhelmed. This means the lysosomes become engorged and clogged with the pigmented sludge, effectively overwhelming the cell's internal recycling machinery. When the recycling plant stops working, the rest of the cell starts to choke. The presence of high levels of lipofuscin physically hinders mitochondrial function, slows nutrient transport, and is strongly correlated with the functional decline of these high-cost, non-dividing cells.
Okay, so if lipofuscin represents the sludge clogging the cell's internal systems, the other major waste product, advanced glycation end products, AGEs, represents the hardening of the structural tissue itself.
That analogy holds perfectly. AGEs are formed through a non-enzymatic reaction called the Maillard reaction, where sugars like glucose react spontaneously with proteins or lipids. Over time, these glycated products become intensely cross-linked and hardened.
Like caramelization.
Think of it as spontaneous caramelization happening within your tissues. Yes, this process drastically reduces the functional elasticity of structural proteins like collagen and elastin.
And this is a huge problem for the cardiovascular system.
A critical problem. When AGs build up in the walls of blood vessels and arteries, they cause the tissue to stiffen and become brittle. This loss of elasticity, arterial stiffness, is a key driver of hypertension and cardiovascular failure. It chokes the cell's ability to communicate, transport, and function efficiently. These two forms of accumulation are the inescapable physical manifestation of the selection shadow, defining the failure point of the human system.
We've established that mortality is tied to the energy bargain and the subsequent accumulation of damage in post-mitotic cells. But the sources provide crucial evidence that this fate is not an immutable physical necessity.
No, not at all.
We could look at organisms that have evolved specific workarounds to the mitochondrial tax, proving that aging is a regulated biological process, not just a result of passive decay.
These anomalies offer the fundamental proof of concept for modern biogerontology. I mean, if they can bypass the tax, we should be able to engineer our way out of it. And the definitive example is the immortal jellyfish, Turritopsis dohrnii.
The one that can age in reverse.
That's the one. Its mechanism is widely known as a biological wonder. It can perform a complete factory reset. When the adult medusa faces environmental stress, starvation, extreme temperature, or injury, it undergoes a process called transdifferentiation, reverting its fully differentiated adult cells back into a cluster of juvenile polyps.
So it's not just healing, it's going back in time.
It is cycling backward in biological time, restoring its full juvenile potential.
That is an astonishing feat. What are the key molecular levers that allow this sophisticated, targeted biological time travel?
Well, the sources point to complex genetic orchestration, but two mechanisms are central. First, they dramatically upregulate the RT3 gene. RT3 is a sirtuin that functions within the mitochondrion itself to optimize fuel consumption, making the energy engine run significantly cleaner and more efficiently, thereby reducing the initial ROS load.
So it's a cleaner burning engine to start with.
Exactly. And second, and most importantly for the reset, the transdifferentiation process requires intense activation of autophagy. The jellyfish cell triggers a systematic process of self-eating where it aggressively identifies, isolates, consumes, and recycles virtually all of its existing damaged cellular components, waste, and accumulated lipofuscin.
A total spring clean.
A total clean-out.
Yeah.
By cleaning house so thoroughly and reverting the cell's epigenetic profile to its juvenile state, the lineage becomes functionally new again. The jellyfish doesn't avoid the oxygen tax, it just employs an unparalleled high-fidelity cellular waste management system, a system that evolution deemed too expensive for us to run permanently after reproduction.
And contrasting with the jellyfish's intermittent reset, we have the hydra, which maintains its functional immortality through continuous renewal. It demonstrates a different strategic avoidance of the decay problem.
The hydra strategy is one of complete cellular turnover. The body is composed almost entirely of highly proliferative stem cells. The organism replaces its body cells so frequently that the accumulated mitochondrial waste, the lipofuscin, and the damage never reaches the critical concentration required to cause functional decline or disease.
So any damaged cell just gets replaced.
Instantly, any cell that starts to accumulate damage is simply replaced by a pristine new one.
That's amazing.
This is a powerful demonstration of continuous maintenance negating the decay caused by the oxygen tax. It's often used in philosophical discussions as a living example of the ship of Theseus paradox applied to biology. If every component of the body is continuously replaced, does the organism ever truly grow old?
And biologically, the answer is no.
The answer is no. It proves that extreme longevity is a matter of resource allocation and maintenance fidelity.
So by analyzing these anomalies and understanding the selection shadow, modern biogerontology has drawn a clear conclusion.
It has. If aging is a software switch prioritized by evolution to stop maintaining the soma after reproduction, then the goal is to trick the human cell into believing it needs a massive, continuous repair budget. We're essentially trying to rewrite the evolutionary code that prioritizes reproduction over permanence.
Okay, so this means targeting metabolic programming, the internal gauges that signal whether the cell should be in growth mode or maintenance mode. The first major target, detailed in the sources, is the mTOR pathway.
mTOR.
mTOR, or the mammalian target of rapamycin, is the cell's master regulator of nutrient abundance. When nutrients are plentiful and growth factors are signaling, mTOR's complex 1, or mTORC1, signals abundance, telling the cell, resources are abundant, grow, divide, and reproduce.
It's the go signal.
It is. And if we inhibit this pathway using compounds like rapamycin, which is currently approved for other uses but extensively studied for longevity, we signal to the cell a state of perceived scarcity.
The cell interprets this resource deprivation as a necessary survival moment. It shifts its metabolism from anabolic growth toward catabolic maintenance, entering a dedicated maintenance and repair mode.
And that kicks off a cleanup process.
A huge one. This switch activates essential cleanup processes, including the sophisticated machinery of autophagy. By forcing this shift, we are effectively overriding the selection shadow's decay mandate, compelling the cell to budget more energy toward high-fidelity repairs, such as clearing out early cellular debris.
So we can turn down the growth signal. Can we also turn up the maintenance signal?
Yeah, that's the other side of the coin. We target the opposing pathway, the AMPK pathway. AMPK, or AMP-activated protein kinase, is activated when the ratio of AMP, a low-energy signal, to ATP, a high-energy signal, increases. We use hormetic mimetic signaling compounds that mimic beneficial stress, or hormesis, to activate it.
This is where drugs like metformin come into the discussion, even though it's primarily an anti-diabetic drug. How does it act as a hormetic mimetic?
Metformin signals artificial energy scarcity by mildly inhibiting complex I of the mitochondrial ETC, similar to the low-level stress achieved through intense exercise or fasting. This small, non-damaging stress activates the LKB1 enzyme, which in turn activates AMPK.
And the cell thinks it's starving a little bit.
It thinks it needs to be careful. By signaling this scarcity, the cell is forced to budget carefully, shifting resources toward survival, prioritizing maintenance, and forcing the allocation of energy toward addressing damage caused by the oxygen tax. It essentially tricks the cell into activating its survival circuits.
Beyond manipulating the cell's immediate risk assessment, we are moving toward radical, full reboot strategies that aim to fully reset the internal programming, specifically through epigenetic reprogramming.
This is a really cutting-edge stuff. This involves the potential application of the Yamanaka factors. These 4 transcription factors can revert specialized cells back into induced pluripotent stem cells, a complete juvenile, undifferentiated state.
A total reset.
A total reset. The theoretical promise is to selectively apply these factors to our post-mitotic cells, the neurons and cardiomyocytes, to reset their epigenetic clock to a juvenile state. When a cell is young, the repair instructions are fully active. As we age, these high-fidelity repair genes are epigenetically silenced. By carefully and partially applying these factors in a pulsed or transient manner, the goal is to erase the accumulated aging signatures without risking the full dedifferentiation that could lead to tumor formation.
So it's a very delicate process.
Extremely. It is a precise attempt to restore the software of youth, providing the cell with the original, undamaged blueprint for maintenance.
But all this sophisticated signaling and rebooting requires vast amounts of metabolic capital. You can flip the switch, but if the bank account is empty, the repair crew can't buy materials. That brings us back to the necessary funding mechanism, NAD plus P.
Yes, NAD plus ESA, or nicotinamide adenine dinucleotide. It's an absolutely critical coenzyme for almost all cellular energy and repair processes. It serves as the direct fuel source for key maintenance enzymes, notably the sirtuins like SIRT1, 3, and 6, which are involved in DNA repair and maintenance, and PRPs, which are crucial for detecting and patching DNA strand breaks caused by ROS.
And our levels of NAD plus drop as we get older?
They decline precipitously, yes. Yeah. This means the cells become metabolically starved of repair fuel. Even if we successfully trigger the maintenance signal via mTOR inhibition or AMPK activation, the cell physically cannot execute the high-fidelity repairs required to overcome the accumulated damage from the oxygen tax because it lacks the necessary substrate.
So keeping those levels up is key.
Optimizing NAD+ levels is therefore essential. It provides the energetic funding necessary for all other modern interventions to succeed at scale.
Okay, so we've established that aging is a strategic choice, the software, and we are developing ways to trick that software. But for our post-mitotic cells, which are choked with decades of accumulated physical waste, the lipofuscin and the AGEs, simply flipping the maintenance switch isn't enough. We need to move from software interventions to aggressive hardware cleanup.
Yes, you have to take out the trash that's already there. We must first acknowledge the cell's own attempt at hardware cleanup. Hormesis. As we mentioned, beneficial stresses like fasting, intense exercise, or thermal shock trigger defensive responses that engage chaperone proteins.
The quality control mechanics.
Exactly. Chaperone proteins function as internal quality control mechanics. Their job is to help refold damaged or misfolded proteins back into their correct functional shape. This is an essential high-fidelity cleaning process that is boosted by mild stress.
But there's a limit.
Oh, there's a hard limit. The limitation here is absolute. Hormesis can only handle relatively new or refoldable waste. It cannot touch the deep-seated, intractable, cross-linked aggregates like lipofuscin or the advanced hardened AG structures. For that, we need to bypass nature entirely.
Here's where it gets really interesting. We are now discussing the cutting edge of technological intervention, using computational power and AI-driven molecular design to create tools with atomic-level precision. This is where platforms like AlphaFold come into play.
A total game changer for biology.
These computational platforms are revolutionizing drug and enzyme design by predicting protein structures with incredible accuracy.
Yeah.
They're being used to design de novo proteins engineered from scratch, customized to perform tasks that evolution never bothered to develop.
Right.
The goal is to create specialized tools that act as atomic precision molecular janitors.
This is the radical concept of engineering a protein specifically to solve the waste management problem the selection shadow have ignored. Researchers are attempting to design a synthetic enzyme, a lipofuscinase, that possesses an active site geometry capable of chemically breaking the extremely complex cross-linked bonds found in the intractable lipofuscin aggregate.
And the challenge there must be immense.
Immense is an understatement. The AI must design a protein that can not only dock precisely with the lipofuscin, which has a highly variable structure, but also operate inside the hostile acidic environment of the lysosome.
And it has to be precise.
And crucially, it must target the waste without causing collateral damage to the healthy surrounding cell structures. If successful, this represents the ultimate hardware fix for decades of accumulated waste in non-replaceable neurons.
So if the long-term goal is the custom-designed molecular shredder for lipofuscin, what are the current, more immediate hardware interventions targeting existing cellular waste?
Well, we have molecular scavengers designed to remove specific lipid-based junk. One promising category is cyclodextrins.
Which, as we mentioned, are essentially molecular baskets.
Precisely. They are ring-shaped sugar molecules used to chemically package and sequester difficult, often lipid-based waste products from inside the cell that the lysosomes cannot handle. They work essentially like targeted cellular garbage trucks, binding to the lipid aggregates and facilitating their removal from the cell.
And this could apply to a lot of age-related diseases.
Absolutely. This is particularly relevant for diseases where lipid or cholesterol aggregation causes cellular toxicity, offering a potential path to clear some components of the sludge.
Okay, so that's clearing the existing waste. What about preventing it from forming in the first place?
That's the ultimate goal. The final, forward-looking hardware intervention targets the very root of the oxygen tax, the leaky mitochondrial engine. We are moving toward neutralizing the ROS leak at its source.
Oh.
Researchers are exploring advanced gene therapies designed to install permanent, self-cleaning catalytic antioxidants directly onto the inner mitochondrial membrane where the superoxide radicals are generated.
This sounds like installing a high-performance filter directly onto the exhaust manifold of the cell's engine.
That is the perfect analogy. Instead of relying on generalized cellular antioxidants like vitamin C or E, which are often inefficiently delivered to the mitochondrion, these engineered solutions involve synthetic enzymes, such as superoxide dismutase mimetics. These enzymes are designed to instantly neutralize the superoxide radical as soon as it forms, converting it harmlessly into oxygen and hydrogen peroxide before it has a chance to react with end-damaged DNA or lipids. If successful, this intervention would fundamentally undo the toxic legacy of the endosymbiotic merger without sacrificing the enormous 15-fold energy advantage that defines complex life.
So what does this all mean? We have meticulously traced mortality back to a single, profound evolutionary trade. We traded the simple, continuous immortality of low-energy anaerobic life for the high-energy sentient existence powered by the mitochondria.
That's the bargain.
Mortality, then, is not a metaphysical mandate. It is the inevitable rust of that energy production, a price evolution was perfectly happy to pay because it prioritized genetic propagation over individual permanence.
And what defines the current moment in human biogerontology is the realization that this decay is not destiny. It is a constraint defined by the strategic limits of the selection shadow and the specific molecular requirements of maintaining a stable, post-mitotic identity.
So we know the rules of the game now.
We do. By understanding these precise evolutionary constraints, the why and how of our decay, we are moving past generalized treatments and are developing the precise molecular tools, the software switches, the epigenetic reboots, and the AI-designed hardware required to bypass the limitations of our own history.
The pursuit of longevity is not fundamentally about fighting a vague, pre-programmed clock. It's about undoing a multi-billion-year-old bargain that define the very terms of complex life itself. We are, for the first time, technologically capable of rebudgeting our cells' energy resources and restoring the high-fidelity repair mechanisms that evolution deemed too expensive to run permanently in a post-reproductive body.
And that leaves us with a profound, final, provocative thought for you to carry forward. If mortality is simply the unmaintained rust resulting from our high-energy engine, and we develop the tools, the molecular janitors, and the permanent internal filters to fully halt that corrosion through targeted molecular engineering, does the concept of a natural human lifespan cease to be a biological necessity and become instead purely an engineering problem that we can solve?